everyone to our first webinar of the fall series. So Advances in Variant Interpretation with Dr. Terra Brannon. So Dr. Brannon received her PhD in molecular biology from the University of Colorado School of Medicine and performed her postdoctoral studies at the University of California, San Diego. Dr. Brennan's academic research broadly focused on RNA biology, including the development of sequencing approaches to study RNA composition and patient-derived stem cell models to study neurodevelopmental disorders arising from variants in RNA metabolism genes. Terra joined Ambry Genetics in 2018 as a variant scientist, where she focused on using RNA data in the interpretation of complex variants in cancer predisposition genes. She's currently a supervisor on the variant assessment team, where she manages a team of variant scientists and RNA analysts in the interpretation of complex variants in both rare disease and oncology. She's a member of several variant interpretation consortia, including the ClinGen Hereditary Breast, Ovarian, and Pancreatic Cancer VSEP and the ClinGen CDH1 VSEP. And we all know that CDH1 is my favorite gene. So I'm very excited to introduce Dr. Brannon. And then when you're ready, you can go ahead and take it away.
Awesome. Thank you so much for that introduction. And thank you all for giving me this opportunity to talk about one of my most favorite things in the world, which is variant interpretation. See, so to start off, I need to disclose that I'm a full-time salary employee at Ambry Genetics. Okay, so let's go over some learning objectives. By the end of this talk, my hope is that you should be able to describe the ways in which variant interpretation is evolving, differentiate the roles of the ClinGen sequence variant interpretation working groups and variant curation expert panels in the advancement of variant interpretation, and recognize that advances in variant interpretation improve variant classifications. Okay, so before we begin, just wanted a big picture talk about what variant interpretation is. So variant interpretation is a weighted system where a curator evaluates all the available evidence. And so does this evidence support a benign impact or does it support a pathogenic impact? And this evidence can include things like population frequency, phenotype data, in silico, functional data, etc. And so all of these pieces of data are given a weight and weighed together. And you ask the question, is this weighing more strongly in the benign direction or more strongly in the pathogenic? And then eventually you come up with a final classification. Most commonly used is the 5-tier classification system, where you have a range of classifications based on confidence, right? Where you have a benign call is the strongest, confidence for a benign impact, all the way up to pathogenic, which is the strongest confidence for a pathogenic impact. And then notably, you have this indeterminate range with a VUS or variant of uncertain significance, where there really is not enough evidence to make a call one direction or the other. So before we dive into variant interpretation, Before we dive into variant interpretation, it's important to highlight that variant interpretation cannot occur without establishing a gene-disease association first. I think somebody is unmuted. Oh. But if it's a question, feel free to speak up. So in other words, when we evaluate a variant for pathogenicity, what is the specific disorder or disorders that this gene is associated with? And how confident are we that variants in this gene cause disease? And so that's where gene disease validity comes into play. So similar to variant interpretation, gene disease validity evaluates the evidence supporting or refuting a gene's association with a specific disorder. And so genes with limited or disputed evidence, so those below this gray bar right here, are considered uncharacterized, where genes with moderate or strong evidence or definitive evidence above this gray bar here are considered characterized. And so a gene's ranking in GDV has important implications for variant interpretation. So only characterized genes can receive the full 5-tier classification system, while uncharacterized genes are almost always VUS because there really aren't evidence codes that can be applied to them. Okay, so now that we got that out of the way, let's take a closer look at variant interpretation guidelines. And so you've probably all seen multiple models for variant interpretation. Most broadly adopted are the 2015 ACMG guidelines, which I'm showing you on the left here. And so in this system, there are multiple types of evidence, including population data, computational, functional, clinical data, as shown on this very first column here. And so each line of evidence has its own code and characterizes either benign evidence, shown on the left of the chart, or pathogenic evidence, shown on the right, with the strongest benign evidence all the way on the left here in purple, and the strongest pathogenic all the way on the right in red, and decreasing weight as you move away from these two ends. So there can be multiple lines of evidence that receive the same weight. So for example, you can see under the supporting, pathogenic supporting right here, PP, in this green column, you can see that there's multiple different evidence codes here. So there's in silico, PP3, missense constraint, PP2, segregation, and PP1. And so I don't want you to worry too much about each of these codes, but know that codes starting with a B are the benign codes, and codes starting with a P are pathogenic. And the second letter in the code denotes the strength, with BS being benign strong, BP being benign supporting, PP pathogenic supporting, etc. Okay, so after a curator has collected all the data for a variant and ascribed a code and weight, the combination of codes are used to come up with a final classification in the 5-tiered system. And so one thing to note here is that multiple different combination of codes can lead to the same classification. So for example, if you're looking at likely pathogenic right here, you can see that 4 supporting and 1 moderate equals likely pathogenic, but also a very strong and 1 moderate also equals a likely pathogenic. So different combinations can lead to the same classification. And so these initial guidelines were developed really to create sort of a standardized terminology and framework to classify variants. And one thing that the authors did note is that this was sort of written as a one-size-fits-all approach, and encouraged that there should be revisions of this framework, including the development of gene and disease-specific guidelines over time. And so that's sort of what the focus of my talk is how has variant interpretation evolved since 2015? Okay, so the bulk of the advancements that I'm going to focus on today come from what's known as the Clinical Genome Resource, or ClinGen for short. And so this coalition consists of the Sequence Variant Interpretation Working Group on the right here, as well as disease-specific working groups here on the left. And so one disease-specific working group that I will be referencing frequently today are the Variant Curation Expert Panels, or VSEPs for short. And so the SVI develops recommendations to variant guidelines to improve consistency. And then the VSEPs take these general guidelines to generate gene-specific variant interpretation guidelines. And so the topics that the SVI focuses on are identified from feedback either from clinical labs, as well as the GSEPs and the VSEPs that exist within the ClinGen domain. All right, so there's really been quite a few publications that have come out from the SVI refining specific codes from the ACMG 2015 guidelines, and there's really just too many to discuss in one talk. So the purposes of today's talk, I'm going to focus on PVS1 and functional studies, which are coded under BS3 and PS3, shown here. And so I chose these 2 codes because I feel that in my personal experience, differences in how laboratories use these codes are often a significant source of discrepancy in variant classification. And I think that's also likely why the SVI kind of chose to focus on these ones first. And in addition, the ClinGen recommendations for the usage of these codes have been out for a few years now, and so we can already start to see how they're being incorporated into gene-specific guidelines and VSEPs. And so I'll also touch briefly on in silico, which is under PP3 and BP4, and kind of based on newer guidance from the SVI that really hasn't been incorporated into VSEPs, I'll take a moment to kind of speculate how we will start to see these recommendations incorporated into new guidelines. And then lastly, after all of that, I will give you some insights into where the future state of variant interpretation is headed, and then we will end with a game because we're fun. So, okay, so starting with PBS1, which carries a very strong pathogenic weight, basically the highest weight you can give in a pathogenic direction. And so in the original guidelines, this was described as a null variant in a gene where loss of function is a known mechanism of disease. So null variants are those variants that we expect will not result in a protein being produced. And so this includes things like full and partial gene deletions, nonsense frameshift variants, variants impacting the +1, -1, or +2, -2 position of the intron. Maybe you've heard them referred to as canonical splice sites, gross duplications, and initiation codon variants. And so in the original guidelines, this was granted, these variants were just granted a very strong pathogenic weight based on the variant type, which can basically make these a VLP without very much other data. But they did hint at some cautions where you may not want to apply PBS1, including in-frame impacts, variants at the very C terminus of the protein, which would escape NMD and potentially produce a stable functional protein, and considerations for mechanism of disease, right? If the mechanism of disease is gain of function, then you wouldn't be applying PBS1. So then in 2018, the SVI generated a framework that incorporated these cautions into it and built out decision trees around possible impact of a particular variant to the protein, and allowed now for a weighted scale where PBS1 could be reduced in situations where it was uncertain if that variant would result in a null impact. And now variant curation expert panels have sort of taken this framework and are using it to define critical regions of the protein for their gene of interest and to provide the final weights for various variant types, and may increase or decrease their PBS1 recommendation strengths from what was recommended from the SVI. So let's take a closer look at this. Okay, so here I'm showing you the updated framework for PBS1 provided by the SVI. Essentially, they created decision trees for every eligible variant type. Here I'm just showing you non-censored frameshifts on the top and GTH canonical splice sites, but similar decision trees were made for other variant types, including gross deletions, duplications, and initiation codons. And so basically, these decision trees take into consideration, sort of as a first step, whether or not NMD is predicted. And if it's not predicted to occur, then you're sort of taking into considerations, is the region critical to protein function? If it's not known to be critical to protein function, then how much of the protein is removed? Is it arbitrarily sort of set here at 10%? And so hopefully you can see that NMD escaping variants that remove less than 10% of the protein receive PBS1 moderate strengths. And so what's important about this is that these will need pretty significant additional evidence to get to VLP, which makes sense, since it isn't clear that such small impacts will be disruptive. And so I think this decision tree also really highlights how important VSEPs are. How do you determine if a region of the protein is critical? Making this call kind of takes substantial gene expertise that may not be available to, a curator who curates, variants in all different kinds of genes and not maybe specializing in one single gene. So let's take a look at how the modified ClinGen guidelines can be used to develop gene and disease-specific guidelines. So here I'm showing you a recent publication from the Hereditary Breast, Ovarian, Pancreatic Variant Curation Expert Panel, or HPOP for short, which developed rules, gene-specific guidelines for variant classifications in the ATM gene. And so as part of this, the VSEP developed PBS1 decision trees for ATM. So essentially what they had to do is take this really, really large gene and research the various functional domains. And so I'm showing you kind of the domains of the ATM gene. You can see ATM gene, you can see that there's quite a few. And they had to research them and provide guidance for what weights can be applied to disruptions in this gene and like what regions are considered critical. And so on the bottom here, I'm showing you one of the decision trees developed by this VSEP for non-sensor frameshifts. They did the full spectrum of variant types, but I don't feel the need to show them all. But similar to the SVI recommendations, the VSEP elected to give PVS1 to NMD predicted variants, which isn't surprising. Those are pretty safe variants to apply PVS1 to. But the VSEP did elect to establish the C terminus of ATM up to amino acid 3047 as a critical region of the protein. And so they upgraded weight for NMD escaping variants in this region to PVS1 from the original PVS1 strong recommendation by the SVI. And so essentially these variants are getting the same amount of PVS1 weight as the NMD predicted variants, because that region of the protein was determined to be so critical. And then finally, the VSEP couldn't find any evidence that sort of the last 10 amino acids of the protein were critical to function. And so they downweighted this region to no PVS1 weight. So no PVS1 weight granted, which was a drop down from the moderate recommended by the original guidelines. And so hopefully this kind of shows you how, the SVI provides a framework, and then these VSEPs can kind of take that framework and deviate from them as, as they feel is needed for their gene of interest. Okay, so Rebecca, I'm so happy to hear that you love CDH1. I did put a CDH1 variant in here. So let's kind of try classifying a CDH1 variant together using PVS1. So first of all, hopefully you remember that there's no variant assessment if the gene isn't characterized. So let's take a look at that first. So ClinGen has really a great resource for looking up gene disease validity. And for CDH1, you can see that there's a definitive association with diffuse gastric and lobular breast cancer syndrome. And the mode of inheritance is autosomal dominant. So first, we'll just classify this variant using the 2015 guidelines. And then we'll compare that classification to what we would get with the updated PVS1 framework as a comparison. Okay, so first, looking at the population data, this particular variant is absent from NOMAD, which is a large general population database. So we can apply this code PM2, which stands for rarity in the general population. Clinical data we have, the variant was detected in a female in her 40s with no personal history of cancer and a family history of breast and prostate cancer, but does not meet any criteria for PS4, which is the code given for clinical overlap. So no weight is given there. The variant type, the variant impacts the -2 position of the canonical acceptor site. So this is one of those canonical splicing variants that are granted PVS1. In silico, using a splice AI predictor that shows 100% probability of native acceptor loss and 97% probability of a cryptic acceptor site gain. However, you're not allowed to apply PP3 whenever you're using PVS1. So no weight for in silico. And then there's really no other data for this variant. It hasn't been reported in the literature or any other databases. So the final classification using those original guidelines comes out to VLP. All right, let's try using the PVS1 framework for this one. So in this first step right here, there's really sort of two decision points that they kind of lump into one. So first is, what is the predicting splicing impact? Is it exon skipping or cryptic splice site usage? And so in general, if there is a cryptic splice site predicted, you assume usage of the cryptic splice site, otherwise you assume exon skipping. So looking at our computational tool to review for cryptic sites, we see we have a 97% probability of an acceptor site gain, which is quite strong, right? 100% probability of loss, 97% probability of gain. And so as a reference, data from the SVI suggests that scores greater than 20% are likely to impact splicing. So this is definitely a significant acceptor gain. So we can consider the cryptic acceptor site as the predicted impact. Okay, our next decision point is to determine if use of the cryptic splice site disrupts the breeding frame. Okay, so if this acceptor gain were used, it would result in an in-frame deletion of only 3 amino acids. So we can answer no to this question since the frame is maintained and eliminate these top 2 paths. Okay, our next decision point is to determine if the deleted region is critical to protein function. So as I mentioned earlier, determining critical regions can be a really tricky process. Some of the things that are recommended to consider are, is this a critical domain of the protein? Are there functional studies showing that this region is critical? Are there missense pathogenic variants in the deleted region that would then inform you that the region is clinically significant? Okay, so while these 3 amino acids are contained within the E-cadherin 5 domain, which is, as a domain quite critical to protein function, there really isn't any evidence that these 3 amino acids are part of a critical region within that domain. They're not part of a motif, they're not part of an active site. So not really critically or clearly critical. Okay, there are no functional studies available, and there are no missense pathogenic variants in the region. So based on this evidence, we would probably say it's not clearly critical to protein function, and we go along this path of the role of region and protein function is unknown, eliminating the bottom path. Okay, so the next part of the decision tree again kind of contains 2 decision points in one question. So I'll break it up into those 2 parts. So first is to determine if loss of function variants in this exon are frequent in the general population. And so the reason why it's recommended to look at that is because the presence of high frequency loss of function variants in an exon may indicate that that exon isn't clinically significant. And if it's not clinically significant, we wouldn't want to be applying PBS1. So we can review population databases such as Nomad and see that there's only 1 loss of function variant in this exon in the general population, but it only contains 1 allele, which is still considered quite rare. So we can answer no to this question. Okay, so then the next decision is to determine if the exon is excluded from biologically relevant transcripts. And so this is sort of similar to that previous question, basically just making sure that the exon is clinically significant. If it's not clinically significant, then there may be other highly expressed transcripts that lack this exon. So we can review numerous transcript databases and determine that there's no well-supported transcript missing this exon. And so we can answer no to this question and exclude that top path, getting closer to the end. Okay, so the final decision point asks how much of the protein is removed, specifically if greater than 10% is removed. Okay, so there were only 3 amino acids removed, and the total protein size is 882 amino acids. So this leads to a 0.34% impact, quite an extremely small impact. So we can answer no to this question and exclude the top path and follow the less than 10% protein path. Okay, so making the last few moves in the decision tree, we end up with a final PBS1 weight of moderate. Okay, so how does that impact our classification? Now with only PBS1 moderate and PM2, the final classification comes out to VUS. And so I'll note that this particular variant and other variants at the same splice site with similar acceptor gain predictions have not yet been seen in any patient with a personal or family history consistent with HDGC. So I think VUS makes perfect sense for this particular variant.
Terra, can I ask a question?
Yeah.
So when you were doing you were calculating the cryptic acceptor site gain, you got like 97%. What is the software tool you're doing to make those predictions?
Yeah, I used Splice AI.
Okay, thanks.
Yeah, and I believe like other predictor programs it comes up with as well, the same gain. All right, so next I'm going to move on to PS3 and BS3. codes. So as written in the 2015 guidelines, this code is used for a well-established in vitro or in vivo functional study showing a damaging for PS3 or no damaging BS3 effect on protein function or splicing. As this code was originally written, it was to include both protein and RNA functional studies, but for the purposes of today's talk, I'm just going to consider protein function. Okay, so in the original ACMG guidelines, they recommend a strong pathogenic evidence or strong benign evidence for functional studies, which you can imagine really, I mean, that carries a lot of classification evidence that can very much drive a classification there. And so in 2019, the SBI in this Burnich publication developed a full scale of weights available for functional studies and determined these based on how well the assay predicts pathogenicity. So they would use something like odds path calculation to determine how well the particular assay predicts a benign from a pathogenic impact and vice versa. And we'll go into more detail in a second on that. And so now in current state, VSEPs are reviewing functional studies for their genes and determining what is the appropriate type of functional study and determining the final weights that should be ascribed to these studies, and also developing recommendations on how to combine evidence from multiple different assays or how to deal when there's conflicting data from multiple different assays. So let's take a closer look at this. But before I dive more into the SBI guidelines, I think it's important to highlight why this type of framework is needed. And so with the advent of high throughput gene editing approaches, such as CRISPR, and the decreased cost in sequencing, we're starting to see more and more large throughput functional studies. And so these are commonly referred to as multiplexed analysis of variant effects, or MAVES for short. And they're essentially capable of testing pretty much every single permutation possible in a gene. And so we're starting to see functional data more and more in variant classification. But the question really is like, how do we assign weight to these studies? How do we evaluate how well they predict pathogenicity? And so that's sort of where the Burnich guidelines come into play. Okay, so here I'm showing you the framework that they came up with, which provides a kind of general decision tree to how to evaluate a functional study for weight. And it considers things like how well the assay models the disease mechanism, the use of controls and replicates. And so for the sake of time, I'm not going to go into every single piece of this framework, but there are a few things I do want to highlight. And so first is that if less than 10 variant controls are used, and so a variant control is a variant that is clearly pathogenic or clearly benign, so a classified variant, the weight maxes out at supporting. And so this is really, this is a significantly lower weight than what was originally granted by the original ACMG guidelines, which was strong, right? If you have 11 in total, then you can give moderate. And so then after 11, the authors recommend calculation of what is called an odds path over here. And essentially, this is just a statistical approach that measures the performance of an assay calibrated against known path and benign controls to determine how well the assay predicts pathogenicity. And so based on the number of controls used and their performance, in theory, you could have even a very strong weight in the pathogenic direction, which really highlights sort of the power that MAVE studies can have on variant interpretation. However, I mean, I think similar to, what I was saying with PBS1, you can see that evaluative functional study using this framework requires a significant amount of gene-specific expertise, right? What is the appropriate assay for the gene? What weight should a study be given? And how do you interpret variant effects for multiple assays? How do you interpret things like intermediate activity? And this is sort of where having a VSEP comes into play as being very helpful. Okay, so here I'm showing you a publication from the TP53 VSEP, which developed gene-specific variant classification guidelines for TP53. And essentially what they did is they evaluated the performance of two large throughput functional studies. This CATA one right here, which tested TP53 transactivation and tested over 2,000 variants. And another one, the Giacomelli 2019 study, which tested over 8,000 variants in response to drug toxicity. And then, so the VSEP evaluated how the performance and concordance of these assays and came up with a decision tree for how to apply weight. Okay, so I'm showing you this decision tree over here on the right. So the decision tree kind of considers the impact from both studies. And if they're concordant, then strong weight in the pathogenic direction or strong weight in the benign direction can be given. If they're discordant, no weight can be given. If one assay, for example, shows, the CATA shows a partial function output, weight can still be given, but the weight then is reduced to pathogenic moderate or benign supporting. And then finally, if data is not available from the transactivation study, the CATA study, because you can see it tested fewer variants, then weight can still be given if there's some other assay available that's concordant with Giacomelli. But again, the weight is reduced in this scenario to either moderate, pathogenic moderate, or benign supporting. Okay, so let's go ahead and take a look at an example on how MAVE studies can impact variant classification. So here I'm showing you a missense variant in BRCA2, C.7819A to G, P.T2607P. And so again, reviewing our gene disease validity, you can see that BRCA2 has a definitive association with an autosomal recessive condition, Fanconi anemia, and a definitive association with an autosomal dominant disorder called, which they term as BRCA-related cancer predisposition. So let's go ahead and look at the evidence available to classify this variant in 2021. So population data, the variant is absent from NOMAD, so PM2 applies. Clinical data, the variant is detected in a female with a personal history of breast cancer in her 50s, and she has a family history of 2 sisters with breast cancer before 50, a mother with breast cancer in her 60s, and a maternal grandmother with breast cancer in her 40s. But this does not meet criteria for PS4. In silico Basedell predictors predicting deleterious, so PP3 applies. And then there's really no other data for this variant, it's not reported in the literature or other databases. So classification as of 2021 is of VUS. Okay, so flash forward to 2024, and a new large throughput functional study comes out, which tests 462 variants in BRCA2 and has sufficient variant controls to give a strong pathogenic evidence for non-functional variants. And so this variant is tested in this assay and shows a deleterious impact, a non-functional impact in this HDR assay. And so now we're able to give strong evidence for the functional weight and allows us to come up with a VLP classification. All right. So finally, I just want to take a moment to highlight a fairly recent update in recommendations for the PP3-BP4 in silico code, which can be given to computational predictor programs. And so per the ACMG guidelines, original guidelines, in silico was sort of capped at only a supporting pathogenic or supporting benign evidence. So it really didn't carry a lot of weight in variant classification. However, more recently in 2022, the ClinGen SBI performed a comprehensive analysis of the performance of multiple in silico predictors and calibrated their performance against a classification of over 11,000 ClinVar variants in almost 2,000 different genes to determine how well they predict pathogenicity, sort of similar to what I was kind of discussing for the functional studies. And so based on the performance of these models, they're proposing that in silico can be essentially a weighted scale from very strong benign evidence all the way to strong evidence in the pathogenic direction, depending on the particular in silico tool used and the score that is outputted. So that's sort of shown in this picture right here. So, some perform better in the benign direction, some perform better in the pathogenic direction. And then, you have a range of weights depending on the outputted score here. So what does this mean for VSEPs? So far, I'm not aware of any VSEPs that have yet incorporated these guidelines yet, but I speculate it's something they're going to consider. And so what I speculate is that each VSEP will probably need to determine which in silico tool performs best for their gene of interest and sort of choose that to be used. And they're also going to sort of have to recommend the thresholds and weights for their gene. Did they go off of, you know, the weights that were granted, you know, when we did this large gene study, or did they develop ones based on the performance of, you know, just specifically their gene with their predictor? And so I can really see this having just a huge impact on variant classification, because unlike functional studies, you know, in silico tools are basically available for all variants. And so, you know, I do think that this will be very impactful. Okay, last but not least, I just want to take a moment to discuss briefly on where variant interpretation is headed. So there's kind of been a push to move from that categorical system proposed by the ACMG to a more quantitative continuum, where each code gets a certain amount of points, so positive in the pathogenic direction and negative in the benign direction, and the absolute value of those points is sort of related to how strong the evidence is. And so then each evidence code can be summed up to give a final points, and that final point amount relates to a final, you know, classification over here. And I think this is just such a more intuitive approach to variant assessment. You know, math is much more simple, and you can quickly sum something up rather than having to refer to some kind of chart. So I'm very excited about this move. And so it's also possible that we might start seeing, you know, more than just 5 tiers, and begin to sort of have a range of US classifications from hot, you know, to ice cold and in between. And so part of the reason I'm kind of bringing up the points-based system is that we are about to play a little game where we're going to be doing some variant classifications, and we're going to be using points instead of weights for simplicity. So with that, I will introduce the game.
Oh, dang. Can you hear the sound, Tara?
No, it's playing. Can you not hear it?
No, it's very, very muted. Oh, well.
No, it must be, I need to turn up my, hold on.
No, that's okay.
All right, well, guys, there was supposed to be really fun music.
Yeah, it was the Ghostbuster theme, everyone. It was very cute. So just pretend you heard it. It was very cute.
We're just humming it to ourselves right now. So in the spirit of Halloween, we are going to play a Ghostbusters activity. So the objective here is that we are going to assess the lines of evidence and determine which would be most helpful in reclassifying a variant. I'll present the variant to you in a minute. So how to play. So on the next slide, I have a case where a VUS was identified. And so we'll review the current lines of evidence. And then on the following slide, we will select which additional lines of evidence you would like to review to help reclassify this variant. So I'll just ask that if like you have an idea, maybe come off mic, say what you think, and we can kind of come to a consensus as a group. And so the catch is that I will only allow you to select a line of evidence after you told me why you think that evidence would be helpful in this specific scenario. And then how to win, we need to bust the ghost and find the answer for our patient. And so finding the answer means that you've identified at least 6 classification points for a variant identified in the case, enabling you to report at least a likely pathogenic variant. Oh wait, it just played again for some reason. Okay. So our case is a 30-year-old female with breast cancer, and that was reported to be HER2 positive. There's no family history or pedigree provided, and the clinician ordered a DNA RNA panel. Okay, so what did we identify? We identified this T53 variant, a single amino acid deletion. It was confirmed heterozygous. The GDV is definitive for Li-Fraumeni syndrome and cancer risk. Mechanism of disease, both loss of function and dominant negative, has been reported. Inheritance is autosomal dominant, and there are established diagnostic criteria. The variant is absent from Nomad, and in silico predictions here, SpliceAI shows no splice impact predicted, and Provian is showing a deleterious impact. So at initial assessment, we have 1 point towards PP3 for the in silico predictions. We have 1 point PS4 phenotype criteria for being detected in a family meeting Champray criteria in a published cohort. Of note, there's also some functional data in this publication, but it's never been reviewed or used towards evidence. And then we also have 0.5 points towards PS4 because we have a proband with breast cancer less than 40, that's HER2 positive. So we have a total of 2.5 points, and we're currently a VUS. Let's see if we can get to 6 points to reach VLP. Okay, so what are our options? What would you do? We can request a structural analysis. We could reach out to the provider to try to get more information. We could perform RNA functional studies. We could review the gene disease validity. We could request a clinical lab data share. We could interpret the functional studies. We could request family studies. We could interpret internal RNA data, but in order to do that, we first have to do the RNA functional studies. And then we could also review online databases. So does anybody have a strong feeling of what they would like to do first?
Can we review the online databases? That's might be maybe easiest.
Okay. Does anybody disagree with that? Okay, let's do it. Oh, I don't know why it happened like this, but okay, so basically we review cancerhotspots.org and IARC databases, and these databases actually only are allowed for missense variants. They don't include the in-frame indels, so there's really no help there. So go back to the evidence.
I'm in between structural analysis or RNA. Could tell you why.
Structure. And what would make you want to do one over the other?
Couldn't tell you why. That's just what, my brain states.
Okay, all right. Structural or RNA, does anybody have a strong opinion for one over the other?
RNA.
Okay, you want to do RNA even though there was no splice impact?
Well, I mean, this is a splice variant, right? So those are predictive tools. So then the actual RNA would tell us what's actually happening. I don't know. Okay. I mean, I take it back then.
No, no. Like let's do it. RNA functional studies. Okay. All right, so this mid-exonic missense variant does not have any predicted splice impact, and there's no RDoC calls from the RNA pipeline. So no weight there.
Sorry, Elishay. I steered you wrong.
Do we want to do structural analysis now?
Yeah.
Yeah, okay. Structural analysis. Okay. So the variant is determined to be strongly destabilizing to the protein. So we're able to give 2 points PM5 structure weight now. So we are at 4.5 now. Getting closer.
I'd want to review the functional data.
Right.
Out there.
Sorry, and what makes you want to do the functional data?
Mostly because it doesn't require, like, I'm thinking of some of the other things that are remaining, like provider outreach, family studies are a little bit more difficult. I'd want to review any data that's available first.
Okay, I like that. Does anybody object to that? Okay, let's do it. Okay. the functional data was reviewed. The variant was shown to be inactive with low transactivation activity compared to wild type, and there were sufficient controls used in the study. So 1 point PS3 functional data can be applied. So now we are at 5.5. 5.5. We're so close.
I guess at that point, maybe I would try to see provider outreach just because I know that there was no pedigree submitted. Maybe it'll give us a little bit more data from that perspective.
Yeah, okay. I like that. Does anybody object to that? Okay, great. Let's do it. Okay, so the lab reaches out to the ordering provider to obtain any more information. And the patient speaks with the family members and indicates that their father had died from a sarcoma at age 42, which is very helpful. So now the proban now meets revised Champré criteria. And so with this new info, we can update the previous 0.5 points that we were applying to this proban to a full 1 point PS4 PC phenotype. So we got it. We got our 6 points. Got our extra 0.5 here. Good job, everybody. So let's just take a look at the final classification. So we get 1 point from in silico, 2 points from structure, 1 point from functional studies, and 2 points for phenotype for 2 different probans meeting Champré criteria, our internal proban and now and 1 in the literature. So a total of 6 points for VLP. Yay, good job. All right, so with that, I guess we have, looks like 10 minutes. So if there are any questions about anything in the game or anything that I talked about earlier, now is a great time to let me know.
So if you, Tara, if you do have a splice variant and you do the RNA-seq data and it does show exon skipping, how many points? So would that be PS3? And would you just get +1 for that?
Oh, great question. Okay. So one of the things that I left out of here was the SVI recommendations on how to weigh RNA studies, which is it's new. And so basically they've proposed to now, instead of coding that under PS, to move it under PS1 or BP7. No, PBS1 or BP7. Basically saying now whatever the predicted impact is now a demonstrated impact with RNA studies. So they call it PBS1_RNA. So that way you can separate sort of protein functional from RNA functional. And they also propose sort of how to, classify the result is based off of, it's going to be a weighted scale, similar to, what I've shown you so far with PBS1 based off of the impact on the protein. So first you have to kind of determine, is it going to be damaging the protein? What is the, the PBS1 impact there? And then from there, how good is the RNA study? Is it quantitated? Is it reproducible? Is, the event absent from controls? Is it showing, a substantial impact that we would expect for, in a heterozygote should be 50%, right? 50% impact, or is it showing something like a 10% impact, which we call incomplete, and that might not carry the same sort of risks that something that is a true null allele would carry. And so all of those things are sort of categorized together to come up with a final weight. So the answer is, it just depends.
Sounds complicated.
It is. It's another complicated one, which is why I was like, that one's a whole talk in and of itself.
I'll ask a question, Tara. So this example, I think, is a really good one, talking about how pedigrees and getting that clinical data can be useful. And I know everyone here in Arizona is wonderful and sends in all their records, but could you maybe just shed some light on how useful records are with variant interpretation, or just anything more about that?
Yeah, absolutely. The clinical data is so helpful. For a lot of genes, you can get strong from a single patient, strong level of pathogenic evidence from a single patient meeting criteria. And I can't tell you how often, we classify a variant, it comes out to be VUS, and then they didn't provide any clinical information. They come back and they're like, oh, well, my patient meets, diagnostic criteria. Why is this a VUS? And we're like, well, because this is the only time we've seen it. We only have like in silico or other things, and we don't have the clinical data. So we're really limited, but thank you. And now we can probably give you your VLP. It's one of, I think, the most valuable pieces of information upfront, depending on the gene, of course. And also, it's really nice too when you're like sort of on the fence with a variant and you're like, something isn't looking right, and then it's CDH1 and it's no gastric cancer, person's over 60 or something like that, no family history of it, with like and so that can also sort of tell you like, hey, something doesn't sound right here. Maybe we need to keep this VUS, right? So it helps a lot with interpretation.
I have a kind of similar question. So when we're in clinic and we're looking at variants in ClinVar and there's a discrepancy and labs are talking to each other to sort of clarify that discrepancy, how do those conversations go, or kind of what are the most likely criteria that are being used for maybe reclassifying between laboratories?
Yeah, so it depends. So there's kind of different reasons for discrepancy. And so one can be interpretation, right? Like just differences in your classification scheme, how you weigh evidence, how you, like weigh the functional studies or the functional evidence, that can vary. PVS1, that can vary between laboratories. So things like that, usually you can't reconcile because it's just sort of just a difference in opinions at that point. But things like clinical data can be shared and can, be very significant to impact classification. RNA data can as well. So we always share our RNA data when other labs ask for it to help them get the right classification as well. Usually those things are just internal data that aren't, commonly available to everybody, are the things that are going to be the most impactful in terms of like resolving a discrepancy between laboratories.
Any other questions from anybody?
There's a part of me that's just like vaguely curious about if you clicked on the other stuff, what it would say.
Yeah. Oh wait, here, evidence. Let's see. Review of GDV. So we already have the definitive GDV, so that's not going to help us. Clinical lab data share. Testing lab contacts other clinical labs. The variant has not been detected in any other patients. No help. Usually you can try to get a good sense of whether this will be useful with ClinVar if it's been reported by other labs in ClinVar, and sometimes they'll cite, that they're using clinical data, and so then you're like, oh yeah, this is definitely going to be useful. Family studies. No available living affected relatives and other family members not willing to participate, so no help there. So this is sort of the interpretation of the RNA when we ran the RNA pipeline. And basically the interpretation is there's no predicted splice impact, so let's just treat as the in-frame indel. And we did online database review. So I think that's all of them, right?
Cool. I think that was really cool. I know that like family studies have been kind of something that we're not like pushing as much anymore in like this case, or just in any cases. Are there like specific scenarios where maybe it would be helpful that we should be on the lookout for, or is it really just more focused on like the functional studies and the clinical impact of the patient that we know of?
Yeah. we are also finding that family studies generally aren't fruitful. So we're tending to be much more selective on what we accept at that point. Thinking less about oncology and more on the rare disease spectrum. If there's a possibility, normally exome is going to be a trio, so you would know this information, but if for some reason they didn't send in as a trio, if you can get de novo status, that can have like a huge impact. We could offer that as a family study. If there's like a very specific phenotype in the family, like maybe LFS could be a good one there. And you have multiple living family members that are willing to participate. You might be able to get some segregation data there. But again, it usually ends up being sort of more of like a supporting, line of evidence, and may or may not, push it one way or the other. Yeah, good question.
Any other questions? I know we're coming up on the hour, but any last minute questions? Okay. Well, with that, I want to thank you so much, Tara, for joining us today. We really appreciate your time and educating us. I know this is a topic that many of us very much want to know more. So we really, really appreciate you taking the time and sharing this with us. And then for everybody on the call, the recording will be available on the AGA website if you wanted to review it. Well, yeah, and that's it. So thanks again, Tara. And have